Hello everyone. This video is going to be answering the question, what is extrachromosomal DNA? And this is part one of this series because I think this deserves an in-depth discussion as to its multiple components, because this is a rapidly evolving field of study in the field of cancer biology. And just one more thing is that I'll be referring to extrachromosomal DNA as ecDNA throughout the rest of the video, just for more efficient pronunciation. So let's get right into it. ecDNA has actually been known about for a long time. In fact, they were discovered not too long after the elucidation of the DNA helix itself. But it used to go under a different name, and that was double minute chromosomes, or demons. And I've included an inset of a demon here to illustrate how nefarious we are finding out these demons are in the role of cancer progression. So double minute, or demons, were discovered during karyotyping by pathologists. And karyotyping is the staining of chromosomes in order to visualize them during metaphase of mitosis, when chromosomes have paired up with their homologue and duplicated in anticipation of the division of the cell. And in a bird's eye view, karyotyping can tell where chromosomes have either, amplified through trisomy, or been depleted through monosomy, or undergone fusion, and things of that nature. And in certain tumor cell lines, what we were finding out is that alongside normal chromosomes, which I can highlight here, the classic image of a chromosome, we also saw these little nubs of DNA fragments, which are also duplicating and in little pairs, but were of a very circular nature. And these were demons. They're floating around with normal linear chromosomes. Now, this was actually a shock because normal eukaryotic chromosomes, they replicate in this very tightly controlled linear fashion, in a linear orientation, whereas bacterial cells tend to replicate with little fragments of circular DNA, as we're seeing with these demons. So this introduced a paradigm shift. I mean, what if these cancers, which were of ultimately eukaryotic origin, what if they were operating almost like bacterial chromosomes, which are circular and free-floating and not linear? So, in other words, what if cancers were altering their very genetic architecture in order to progress? So this raised a very important question as to what these demons actually were doing. So alongside karyotyping, as we discover more about cancer genetics, we have discovered the presence of demons in many cancer cell lines, especially advanced ones. And alongside karyotyping, we now have, more refined ways of visualizing chromosomal aberrations, and in fact specific genes that are expressed on chromosomes. So in the top here, we have examples of karyotyping of prostate cancer and Ewing sarcoma. I can highlight where we see some demons here in yellow. Apologies, but you might need to zoom in to see them at a more refined level. But below in figures B and C, in both gastric cancer and colon cancer lines and lung cancer, we can also visualize demons with something called fluorescence in situ hybridization, which is called FISH. And FISH actually selectively fluoresces or highlights specific genes, for example, in our context, oncogenes, that are expressed by different types of chromosomes. So if you pair FISH with a karyotype, you can really tell which genes are being amplified on what chromosomes. And here, as you can see below, we have number one, FGFR2, which is a fibroblast growth receptor oncogene, and it's being expressed on these demons here. And also MYC, the MYC oncogene, which is very useful for the progression of cells through the cell cycle and allowing for their division and proliferation. You can see that's also being highly enriched on demons. Sometimes these are co-expressing. And you can see the same thing in this colon cancer cell line, where we have all these demons that are expressing MYC oncogene expression, very richly expressed. So there's this correlation between oncogenes and demons that has now come to the forefront, and we know that's a fact. Let's go into more depth about oncogene amplification in ecDNA. Now, with advanced techniques such as CRISPR-Cas9, in which we can selectively generate double-stranded breaks around certain genetic targets, we can now linearize or turn these DNA circles into lines that we can analyze with normal electrophoresis and characterize the diverse architecture within ecDNAs. And in certain colorectal cancer cell lines, like colo320DM, we have found that these cells can contain up to 50 to 100 ecDNAs within them, within each cell. And up to 95% of these express oncogenes like MYC, which, as I mentioned earlier, are important for the progression of cells through the cell cycle. Another example is within glioblastoma, or primary brain cancer derived stem cells. These have a very high degree of EGFR transcription, and we found that this isn't due to the normal EGFR on the normal linear chromosomes within these stem cell lines, but they're actually because of ecDNA that contain elevated levels of EGFR. So these are two examples of this phenomenon occurring. So what are some core features of ecDNA? Well, the first is that they have a circular shape, and a circular shape allows them to be insulated from the end protection problem. And the end protection problem is shared by all linear chromosomes. It's that DNA polymerase cannot faithfully replicate the ends of a linear chromosome with each cell division. And so this causes an attrition, or a wear and tear at the ends of the linear chromosomes with each cell division. And if unstopped, it would cause the attrition of the decay of gene products that are coded by the linear chromosomes at the very ends. So this is why we have a transcriptionally silent cap to our chromosomes called a telomere, which sort of staves off that attrition from getting to those gene products. Well, circular chromosomes don't have that problem by their very nature, and so they have increased replicative longevity. And the fact that ecDNA is circular leads to the second point, which is that they lack a centromere. A centromere is a transcriptionally silent kind of center of a chromosome pair, and what it does is that it facilitates the equal division of chromosomes into each daughter cell during each cell division, which is, of course, very useful if you want to have equal and stable chromosome inheritance across all of your progeny cells. And I'll draw a picture of it here. This is what a centromere looks like in the center of this chromosome pair. And so because ecDNA lacks a centromere because of its circular nature, this allows for the rapid copy number amplification across daughter cells, because if you think about it, some ecDNA circles, they can remain unattached and they can float off into one daughter cell, which may inherit too many of one ecDNA copy. And so you can imagine that this causes unstable and high level copy number amplification in certain daughter cells. And then ecDNA, because it's very small, it's very spatially mobile. In comparison to bulky, physically large linear chromosomes, which can't bounce around very often in the nucleus of the cell, ecDNA circles are small, and because of their small nature, they can interact with other genetic elements and with other ecDNA fragments. So this we'll discuss in a second. Finally, ecDNA is composed of very loosely packed chromatin. This loosely packed chromatin is important because it remains more accessible to cellular machinery that actually transcribes and replicates DNA. And so because the gene products within ecDNA are more accessible, this allows for increased oncogenesis and increased oncogene protein formation. Also, this loosely packed chromatin lends itself to a low methylation load. If you know anything about epigenetics, methylation actually silences gene transcription. And so because there's a low methylation load, this only adds on to the easy access to transcription that ecDNA features. This is why patients with ecDNA-rich cancer suffer from worse survival outcomes and chemotherapy resistance over time. So what's another component of circular DNA that can accelerate tumor growth and cancer progression? Well, on the left here, I've illustrated the snake game that everyone's familiar with. And in this game, pretend that the head of the snake is an enhancer element, or an element of DNA that doesn't code for a gene product, but actually enhances the product of another gene. And pretend the tail is where the gene is. So this enhancer is always trying to catch up with the tail, but because of the long nature of the snake, it can't reliably reach it every time. And so this prevents the overproduction of a gene in our normal linear chromosomes. But ecDNAs, because they're circular, on the right here, I've illustrated an Ouroboros, or a snake eating its own tail. This illustrates that the enhancer has caught up with the tail, or the gene. And so this allows, because of this physical proximity between the enhancer and the gene, this allows for increased oncogene protein production within ecDNA. An example of this is with a specific enhancer called PVT1, which normally is found in a different chromosome from MYC the classic oncogene I talked about. But in colon cancer ecDNA, it's actually been found that PVT tends to be fused with MYC in the same circle. And so because of this recursive physical proximity, this allows for increased MYC transcription within ecDNA cell products, and that only increases the oncogenicity of some colon cancers. Now here are some other properties of ecDNA that are derived from kind of the core features that I talked about in the earlier slide. Well, they have mobile transaction elements, and what that means is that enhancers on these ecDNA circles can easily interact in a physical manner with genes on normal linear chromosomes. So you have these enhancers, for example, PVT1, on ecDNA circles, and they can come into proximity with oncogenes on normal linear chromosomes through physical, spatial mobility. An example of that in another cell line is in the gastric cancer SNU16 line, where an ecDNA expressing FGFR2 has enhancers that actually activate a distant ecDNA, which is of a different composition altogether, which expresses MYC. And so that's called a transinactivation because the genes are on two different kind of linear chromosomal paths. And this is through the formation of an ecDNA hub, almost like a juncture where multiple ecDNA fragments can physically approximate with each other and interact with their genetic material. And in fact, some ecDNAs don't even contain coding sequences. They only contain non-coding enhancer element amplicons. And so that only increases their potency and their interactivity via trans interactions to kind of accelerate oncogene expression throughout the genome. The other element is something very interesting called Kyklonic hypermutation. And I'll get into this later. It actually is associated with another gene called ApoBEC3, which we'll get into in a second. So stay tuned. Here in this slide, we have a demonstration of all of these processes illustrated. So in the top left here, you can see how oncogenes can be added to ecDNA circles. On the top right here, you can see how methylation is actually reduced in ecDNA, which allows for greater transcriptivity of genes on that ecDNA fragment. In C, we can see how enhancers can be hijacked and added onto ecDNA, which only increases transcriptivity. And in D, we can see how intermolecular interactions can occur, such as ecDNA circle interactions with the normal chromosome in trans, and ecDNA hubs that form, which are those junctures that I talked about where they can all get together and band up to do nefarious things. And this figure demonstrates how ecDNA can actually be asymmetrically inherited among daughter progeny because, as you can see here, let me turn on the laser pointer again, you can see here that this cell has 3 ecDNA fragments. These duplicate in anticipation of mitosis, but these ecDNA fragments, because they lack a centromere, as I discussed earlier, they don't get symmetrically allocated with the microtubule structures that allow for separation of genetic material equally in normal linear chromosomes. And so you can see in this daughter cell, there's 2 more pairs of ecDNA, and this heterogeneity among daughter cells only increases kind of the copy number amplification that we were talking about earlier. And so this can actually boost oncogene transcription to the top 1% of all transcriptome outputs within the entire genome of the cell. So you can see how that can be a huge problem in cancer progression. And these acentric chromosomes, as I mentioned, this is the reason ultimately for why asymmetric segregation and copy number heterogeneity actually exists. And so let's discuss a little bit more about enhancers can be brought into an ecDNA. This can happen through multiple processes, but oftentimes it happens through a process called chromotripsis, or genetic shattering in Greek. And this is a process in which literally linear chromosomes shatter because of genetic instability in cancers. I'll discuss that in a later video. And that causes the random allocation of genes and non-coding segments of DNA, like enhancer elements, and those are brought together and circularized into ecDNA. Now, this isn't necessarily an active process. I mean, ecDNA doesn't choose to form. It's more of a process that occurs through random DNA ligation from these millions or billions of cell divisions that are happening within tumor cell lines. So random DNA ligation causes the formation of random ecDNA segments. Some of these ecDNA segments have oncogenes. Some of them are asymmetrically segregated into certain daughter cells, which causes copy number heterogeneity. Some of them adopt enhancers. And so there's actually a selective pressure because cancer cell lines with more ecDNA, which have oncogenes and enhancers together, tend to be selected for, or tend to proliferate more rapidly. So you can understand how that's natural selection actually happens, because ultimately natural selection occurs through the asymmetric reproductive quantities of organisms, such as cells. And this allows rapidly growing cancers such as medulloblastoma, glioblastoma, and small cell lung cancer to actually adapt to stress, because more oncogene expression can allow for things such as chemotherapy resistance and more metastasis and things of this nature. And in fact, we're now finding out that some virus-associated cancers, like human papillomavirus-associated oropharyngeal cancer and hepatitis B virus-associated liver cancer, there's actually virus ecDNA hybrids. And as I mentioned, viromes, viral DNA particles, bacterial DNA particles, and ecDNA are actually quite similar in that they have transacting elements, they're small, they're circular, and they lack centromeres. So they're very similar in this regard, and you can observe how they can actually hybridize, which is very interesting. Now, this diagram is actually derived from a very interesting research finding that was discovered recently, and that's that Barrett's esophagus, which is a pre-malignant lesion in your esophagus. Often it's due to high gastric acid exposure or various irritants through the throat that chronically cause cell dysplasia over time. Well, Barrett's esophagus, when it progresses to esophageal adenocarcinoma, which is cancer of the throat, we actually find that the number of ecDNA amplicons actually increases with that progression from pre-malignant to malignancy. So that's very interesting, and it indicates again that either ecDNA is a feature of just malignant cancers by its nature and something that kind of tags along with that process, or that it actually promotes that process. And this figure demonstrates how when we go from pre-cancer, we get a certain complexity of ecDNA, and then we get clonal selection where some cell lines have more ecDNA, some have less. And so this heterogeneity is actually, remember, a feature of the asymmetric segregation of ecDNA fragments into different daughter cells. Now, I mentioned something earlier in the video, and that was that many ecDNAs feature something called Kyklonic hypermutation. What is that? That's a very fancy term. Well, the synonym for that is Kataegis. And 30% of ecDNAs have greater than one katagistic events. And Kataegis actually means something very interesting in Greek. It means thunderstorm. As you can see here, there's a lightning strike here. And what this refers to is a phenomenon that we find that in ecDNAs, there is very intense spatial clustering of hypermutational events. So you can see in this figure here, we see mutation. We see about this area of red here with about 4,000 mutations. And this 4,000 mutation amount, you can see in the y-axis, it is associated with an intermutational distance. And this intermutational distance is very low, because normally you'd expect mutations to be randomly spaced apart and not really clustered together if they're truly occurring randomly. But these Kataegis events, they have a very short intermutational distance and a very high mutational number, which indicates that they're almost like lightning strikes on certain parts of the genome. So why does this happen? I mean, this is not a random process. These can't all be clustered together randomly. So let's discuss that next. We're actually finding now that there's a specific protein or enzyme called apolipoprotein B mRNA editing enzyme catalytic polypeptide 3. I know it's a mouthful. And this is actually a driver of Kyklonic hypermutation and Kataegis. So what does it actually do? Well, ApoBec3, it's actually responsible for cytidine deamination. Remember, the cytidine is one of the core building blocks of DNA, one of the nucleotides of DNA. And cytidine deamination turns it into something called deoxyuridine, which is another word for uracil, another component of uracil. And this causes, as you can imagine, if you turn a C into a U, ultimately that U turns into a T, a thymidine, and then the complement to that nucleotide, which is normally a G, a guanine, has to turn into an A, an adenosine. So you can see how all of this causes a selective mutation. The role of this actually is very interesting. In ApoBec3, normally, it generates antibody diversity because antibodies need to have multi-mutational events in order to kind of generate enough complexity to target every antigen that possibly could ever exist in a human body. And ApoBec3 also prevents retroviral insertion into the human genome. How does it do that? Well, certain retroviruses, as you may recall, like HIV, they insert their genetic material back into our genome. And so to prevent that, ApoBec3 kind of like a lightning strike mutates parts of the retrovirus in order to kind of prevent the genetic material from being transcribed properly and the proteins to be expressed properly. So ApoBec3, it normally targets retroviral genomes. Well, remember what I mentioned, that viral genomes are very similar to ecDNA in that they're circular, they're small, and they lack centromeres. So the hypothesis is that ApoBec3 is actually responsible for targeting these ecDNAs because they look a lot like viruses to our genome. And then what it does is that these mutations don't actually disable ecDNA. In fact, they can actually activate ecDNA by selectively targeting certain oncogenes or enhancers and turning them on, or turning tumor suppressors off. And this kind of messes up the entire kind of oncogenomics and causes acceleration of oncogenesis within these cancers. So very fascinating stuff. And we're actually finding that 41% of all Kyklonic events, I'll highlight this here, are co-occurred with ApoBec3 promoted Kataegis. And these are actually intersect with known cancer genes. So obviously ApoBec3 is not really helping in terms of fanning the flames of oncogenesis within ecDNA. So I hope you guys learned something very interesting, at least a few things from this video. And because this is part 1 of the series, next I'm going to discuss in more in-depth ecDNA formation processes, which can occur through multiple means, some of which I mentioned, like chromotripsis, but there are many others, like excision, non-homologous end joining, and break and fusion bridging. Then I'm going to discuss how ecDNA can actually contribute to chemotherapy resistance and immune evasion privileges within certain cancer cell lines, which are, of course, huge topics of therapeutic targeting nowadays. And then, of course, I'm going to talk about ecDNA-directed therapeutic options, which are coming to the fore. So thank you, and I hope to see you in the next video. And these are my sources. I really hope you guys read some of these. They're very interesting. So thank you.